Welcome to the town square, where we're going to talk about the innate immune system's response to viruses. We've already discussed some mechanisms that the innate immune system can use to defend against viruses outside the cell, like the complement system. But once the virus is inside the cell, well, that's it. Game over. We're screwed. Just kidding. We have millions of years of evolution to thank for keeping us meatbags alive. So let's talk about these intracellular defenses. And just as a reminder, we're talking about the innate immune system, so we're not going to talk about antibodies or B cells or T cells or any of that. Inside the town square, life is normal. It's got normal townsfolk, and they're all wearing badges around their neck that say MHC1, which stands for Medieval Healthy Citizen, of course. For now, let's focus on this baker, who's munching on a random apple pie he found on the ground. And if that pie looks a little odd, that's because it is odd. It's a pie with virus-shaped apples. And our previously normal baker here, who's eating this virus apple pie, represents a cell that's just been invaded by a virus. Possessed even, you might say. He's glowing red and he's baking even more of those virus pies. This means the virus has taken over the host cell, hijacked its machinery, and is now trying to replicate itself. Lesson learned, don't eat random apple pies, my friends. Eventually, these newly made viruses will burst out of the infected cell and go on to infect other cells. And that means that we gotta stop those virus pies from getting into the mouths of our unsuspecting townsfolk, or else we're gonna have more infected people who will infect more people, and you get the idea. The good news is the innate immune system does have a way to interfere with these viruses once they're inside the cell, and we call it the aptly named interferon, which we've symbolized with these interfering ferrets, or if you'd prefer, interferets. The host cell contains pattern recognition receptors, or PRRs, that can sense viral components within its system. When that happens, an intracellular signaling cascade is activated, which causes the virus-infected cell to release interferon alpha and interferon beta, both of which are type 1 interferons. To help you remember this, we've drawn an albino ferret and a black ferret to symbolize interferon alpha and beta, respectively. These type 1 interferons have two primary functions. One, they signal cells to transcribe antiviral enzymes capable of halting protein synthesis. And two, they induce apoptosis. And they do this via two kinds of signaling: autocrine signaling and paracrine signaling. Autocrine signaling, or autobinding, is what happens when the infected cell produces an interferon that then binds to itself. When this happens, it stimulates production of proteins that prevent the virus from further replicating within the cell. Hence why our ferrets here are swiping at the baker and trying to prevent him from baking even more virus pies. Paracrine signaling occurs when the interferon acts as a warning protein. It binds to interferon receptors in neighboring cells to warn them that there are viruses in the area and that they may soon be attacked. So that's like this ferret here, who's screeching at the townsfolk that the baker is making some weird-ass virus pies and they shouldn't eat them. You tell him, interferret. As a result, the neighboring cell is stimulated to mount its own antiviral defense, or in the event that it's infected by a virus, it'll induce apoptosis in itself. In the meantime, however, the cell's regular routine won't be interrupted. It'll do business as usual, like bargain for bread or argue with the blacksmith, until the attack actually occurs. It's important to note that the host cell isn't alone to defend itself. There are two other immune cells that play an important role in the innate immune system defense against viruses. And those are plasmacytoid dendritic cells, or PDCs, and natural killer cells, or NKCs. PDCs, symbolized by Dendrilox, our dendritic cell, are the sentinel cells of the immune system, hence why she's keeping an eye out in the watchtower. She's also hanging out with our sexy troll, which represents toll-like receptors. When a dendritic cell encounters a virus, viral antigens bind to toll-like receptors 7 and 9 on its surface. Those TLRs detect viral RNA and DNA, respectively, and cause the dendritic cells to produce massive amounts of type 1 interferon. And I do really mean massive here. Most of the type 1 interferons detectable in the blood following a viral infection are from dendritic cells, because they're basically interferon factories. That's a lot of ferrets. Furthermore, the released interferons activate natural killer cells, or NKCs, represented by our assassin. NKCs are also on patrol, mainly in the blood, spleen, or liver. And no good assassin works alone, as you can see by this ginger ferret chilling on the NK cell's shoulder. Within the first hours of infection, NK cells are responsible for producing interferon gamma, represented by this ginger ferret. It's also got a 2-shaped tail because it's a type 2 interferon. Interferon gamma has multiple roles. It supports T helper cell differentiation, boosts macrophage function and leukocyte migration to the site of infection, and increases MHC1 expression for better T-cell recognition of infected cells. Speaking of MHC1 expression, remember how we mentioned these MHC1 badges at the beginning of the sketch? Did you notice how the possessed baker's badge is missing? NK cells can induce apoptosis in virus-infected cells, and they do this by detecting whether a cell has decreased or absent MHC class I proteins on their surface. If they are missing those MHC class I proteins, it's usually because that cell has been infected by a virus or is a tumor cell. Either type is bad news bears, so the NKC is going to target them for destruction. Just like this assassin has targeted this baker and his virus pies for assassination because he's not wearing his MHC1 badge. The NKC can detect whether a cell has little to no MHC class I proteins via its inhibitory and activating receptors, which explains why the assassin is flipping their coin. One side is red for inhibition, and the other side is green for activation, specifically activation to kill. When the NKC detects a normal amount of MHC1 on the cell with its inhibitory receptor, the NKC's killing response is inhibited. However, if the NKC detects decrease or absent MHC1 proteins on the cell with its activating receptor, the killing response is activated. Once this happens, there are two ways the NKC can induce apoptosis. The first is the fast-acting cytotoxic pathway, wherein perforin from the NKC forms pores in the cell membrane of the infected cell. So we'll give our assassin a ninja star to perforate these virus pies. These pores create a channel for granzymes, like granzyme B, hence why these ninja stars have a B at their tips. Granzyme B enters and triggers an intracellular signaling cascade that induces apoptosis. If there's something called a fast pathway, then there's probably a slow one too. In this case, the slow-acting cytotoxic pathway occurs when the Fas ligand on the NKC interacts with the death receptor Fas on the infected cell. The death receptor is symbolized by these fasteners on the pie tin, which the assassin has attached to with their Fas ligand hook. When this happens, a disc, that's a death-inducing signaling complex, is created within the cell, which eventually leads to apoptosis, or a pie plate that's been shattered on the floor. Looks like there's going to be a recall on these virus pies, so let's go ahead and do a review before they get pulled from the shelves. The innate immune system is the first line of defense against viral infection. When a cell is infected by a virus, the virus hijacks the cell's machinery and begins to replicate itself. When this happens, the host cell releases interferon alpha and interferon beta, both type 1 interferons, to prevent the virus from further replicating within itself and to also warn neighboring cells about the virus. Two other immune cells are involved in the innate immune response against viruses. The first is the plasmacytoid dendritic cell, which has toll-like receptors on its surface that can bind to viral antigens, causing them to produce large amounts of type 1 interferon. The second cell is the natural killer cell, which produces interferon gamma, a type 2 interferon. The NKC destroys virus-infected cells by determining whether they have decreased or absent MHC1 molecules, which they're able to detect via the NKC's inhibitory and activating receptors. Once the NKC has targeted the virus-infected cell for destruction, it has two primary methods to induce apoptosis. It uses perforin to form pores in the cell membrane so they can insert granzyme B, and the Fas ligand on the NKC can also bind to the death receptor Fas on the infected cell, both of which trigger signaling cascades that lead to cell death. And that's it for the innate immune system's response to viruses. The most important thing to remember here is, don't eat random pies off the floor.